My name is Kevin S. I'm a pediatric neurologist at Vanderbilt Children's Hospital in Nashville, Tennessee. I have several interests in alternating hemiplegia of childhood. I actually direct a clinic for children with AHC at our Children's Hospital for the last couple of years now. We're always interested in seeing more patients to help both increase our understanding of the disorder as well as to help families with the difficult manifestations. I also direct a basic science laboratory that studies AHC as well. And we've been doing that also for the last several years. And one project that we've been working on that's starting to really have some fruition results is we are making stem cells from patients with alternating hemiplegia of childhood. And many people have heard about this technology. This involves taking usually skin cells from patients, growing them in a dish, and then reprogramming them into what we call induced pluripotent stem cells, or iPS cells you may hear about. And this is a real game changer. It allows us to study human cells from actual patients with these disorders who have the known genetic changes in the ATP1A3 gene. And then from those cells, we can do several things that are amazing. We can turn them into brain cells. We can actually make neurons in a dish. We can study what's wrong with those neurons. We can put drugs on them to help discover new therapies. And the other part of it, which is really great too, is if once I have stem cells, they will grow and you can freeze them and thaw them, and we can ship them and share them. So it will become a resource that other scientists around the world will be able to use to further their own research as well. So this is really a time of now August 2016 of amazing progress in AHC and related disorders. But one of the thoughts I have more recently at this meeting is we're getting so much information so quickly about new types of disorders, diseases, different symptoms, different phenotypes, as we say, that are related to the ATP1A3 and other similar genes too. So it's really expanding what we have to think about, what we have to treat, and it's becoming even more challenging given the complexity of this disorder. But one thought that keeps going through my head as I hear other people talk and we discuss AHC related conditions is that we're seeing more and more patients now at younger and younger ages. And I like to remind people who don't think about this like sometimes we do in the pediatric neurology and genetics world, is that DNA sequencing is really such a standard tool now, and it's really how much these things cost. So the ability to get very thorough genetic screens, which is almost a research tool now, or was a research tool just a couple years ago, moving a couple years further in the future, and certainly within 10 years from now, I think everyone will have very complete genome scans done, and then we'll be faced with this wonderful challenge, but also opportunity to have children identified with these gene mutations probably soon after they're born. And then we'll actually have opportunity, I think, to have patients before they even begin to have even begin to have symptoms of AHC, and then to hopefully address them with therapies. So I think the great challenge we have in the field is to, again, one, catalog all the various genetic changes that are part of AHC-related disorders, identify what the proper treatments could be for them, and really consider a world where we can treat before symptoms even occur. That would be a wonderful change and something I'm looking forward to working on. The other challenge, though, in such a great advance is who's going to pay for this. So we, of course, do need more money for research, but more money also for understanding genetic sequencing, how to organize the data. All these things are going through medicine very rapidly right now, but I think, again, the next 5 to 10 years we'll see major changes, and the number of patients with AHC-related disorders will only continue to grow.